Today in this lecture, we will talk about summary of concentrations of different solutes in different tubular segments of the nephron. In short, we will see whether the concentration of a solute in the tubular fluid increases or decreases in any particular segment of the nephron, in the proximal tubule, loop of Henle, distal tubule collecting duct. Basically, whether the concentration of a solute in tubular fluid is increasing or decreasing, it depends on whether that solute is being reabsorbed, more than water being reabsorbed in that segment. If the amount of a solute in a particular segment reabsorbed is more than the amount of water being reabsorbed in that segment, like for example in proximal tubule or loop of Henle, then the concentration of that solute in that particular segment will decrease. But if the amount of water being reabsorbed in any particular segment is more than the amount of that solute being reabsorbed, then the concentration of that solute will increase. Simply speaking, if more water is being reabsorbed as compared to that solute, then the concentration of that solute in tubular segment will increase. But if more solute is being reabsorbed as compared to the amount of water reabsorbed in that segment, then the concentration of that solute in that particular segment of the nephron will decrease. We calculate the concentration of any solute like glucose, proteins, amino acid, creatinine, urea, etc. in the tubular fluid for this diagram by tubular fluid concentration by plasma concentration. For example, concentration of glucose in the proximal tubule divided by concentration of glucose in the plasma. Similarly, concentration of amino acid in the proximal tubule divided by concentration of amino acid in plasma. Similarly, we will take the concentration of urea in all the segments by the concentration of urea in plasma. Now, if the amount of the urea or glucose or proteins or amino acid, if this concentration is increasing, it is increasing above 1, it simply means that that particular solute is not being reabsorbed. But if it is decreasing below 1, then it means it is being rapidly reabsorbed. Sometimes a particular solute may even be secreted. It may even be secreted into the tubule. Some of the solute may not get reabsorbed, rather they may get secreted into the tubule, as we have discussed in detail. That thing also increases the concentration of that particular solute in the tubular segments. So for this figure, we take into account that the concentration of a particular solute in plasma remains the same. It remains constant. So we assume that the concentration of glucose, for example, it remains in plasma the same. It remains the same in plasma. For example, it remains 1. Or we assume that the concentration of urea in the plasma remains the same. For example, it remains the 1. But the concentration of glucose, protein, or urea is changing in the tubular fluid. So as the concentration in plasma remains the same, and this value is coming from concentration in the tubular fluid by concentration in the plasma. So any change in this value above 1 or below 1 shows the change in tubular fluid. And now, if the concentration of glucose in the tubular fluid decreases, for example, initially tubular fluid concentration was 1 and the plasma concentration was 1. For example, concentration of glucose in tubular fluid was 1 and concentration of plasma was also 1. So 1 by 1 is 1. Now, any decrease below 1 will show a decrease in concentration in the tubular fluid. Any decrease below 1 will show decrease in concentration of glucose in the tubular fluid, because we have assumed that the concentration of that particular solute in plasma remains the same. Now let's see, if the concentration of glucose has decreased below 1, the concentration of plasma has not changed. So for example, it has decreased to 0.5 by 1. It will definitely decrease the value of the concentration of glucose, and it will directly show that the concentration of glucose has decreased in the tubular fluid, because the concentration of plasma of that solute in plasma has not changed. Now this figure is also showing one important thing, and it is showing that important substances like glucose, proteins, amino acids which are needed by the body, their concentration falls rapidly, and it falls very rapidly in the proximal tubule, in the very first segment of the nephron. While the concentration of those substances which are not needed by the body and which are bound to be secreted, they are not reabsorbed, rather they are being secreted into the tubule. Their concentration, for example, the concentration of urea, it keeps on increasing in the tubular fluid because it is not being reabsorbed more as compared to the reabsorption of water. So in this diagram, the substances, the solutes which are needed by the body, like glucose, proteins, amino acids, their concentration falls rapidly, and it even falls directly in the very first segment in the proximal tubule. But the concentration of those substances like creatinine, urea, para-aminohippuric acid, it keeps on increasing. Their concentration and the tubular fluid keeps on increasing, and because they are not needed by the body. And this figure is just summarization that how the value of different solutes in different segment is varying. For example, if we take into account the inulin and creatinine, their value keeps on increasing in the proximal tubule, then in the loop of Henle it stays constant, then in the distal tubule their concentration is increasing once again here, and in the collecting tubule it is again increasing. So the concentration throughout the tubule is increasing. While in case of glucose and proteins and amino acids, the things which are very important for the body, their concentration directly come to zero value directly in the proximal tubule, in a normal functioning kidney, in a normal functioning nephrons. So this diagram, this figure, this video is all about the summary of concentration of different solute along the different segments of the nephrons, and the importance of some substances that get reabsorbed, and the secretion of some substances out of the body which are not reabsorbed, and their concentration keeps on increasing in the tubular fluid, and the body wants to get rid of these substances. That's all about the summary of concentration of different solutes and how it changes in the different segments of the nephron. Thanks a lot for watching the video.